Good morning, everyone. All right, I see we're getting some attendees. Welcome. Welcome, everyone. We'll get started in just a minute here. Okay, well, good morning. Thanks so much for joining us. As you enter, please make sure to let us know who you are. Add your name and affiliation in the chat box. You are here today at the third seminar of the Delta Research Awards seminar series. We have a very exciting seminar planned for you today, and today's theme is multi-benefit wetland habitat and Delta food webs. Again, just want to ask that if you are here, please add your name and affiliation into the chat so we can all get to know one another better. You should be all seeing the poll through Zoom, and we're going to have one at the beginning and one at the end of the meeting. So please take a couple of minutes to fill it out. And the purpose is really to continuously improve events like these. So really appreciate your help in advance. We'll just probably wait one more minute for some others to join us. And just so you all know, this seminar and every seminar in the series will be recorded and posted to the Council's YouTube channel in case you want to share this with anyone who's not able to attend or look at it later. We really want everyone to participate in the discussion, ask questions. So please make sure to add your questions in the Q&A box. The questions will actually appear for those who are attending once we decide to answer them live, and then everyone will be able to see them. You can also raise your hand to participate, and I'll promote you so you can to ask your question live. Great. Okay. I think we may as well get started. So thanks again for joining us. First of all, I'd really like to have Eva and Megan introduce themselves really quickly. They are Sea Grant Fellows who are really leading this effort. So go ahead, Eva.
Hi, everybody. Thank you all so much for joining. This has been a really interesting and fun program to try and organize with the team, and I hope you all learned something. I've learned so much, and I think by having our presenters speak to us all directly, we can get a lot out of it. So thanks for joining. Megan?
Yeah, so thanks for joining. Also really glad to be part of this project as well. I'm one of the state fellows working under Don Stern, but yeah, happy to be here, and I'll be controlling the slides. So
Thanks, Megan. Thank you, Eva. A little bit of housekeeping. So again, this seminar is open to the public and is recorded. The format for today is we'll have 30 minutes for each presenter to present and then answer questions. And we do have some additional time at the end for discussion. We do want to encourage everyone to participate and engage. And so please add your questions to the Q&A box. Share your thoughts and reactions in the chat. You can also raise your hand and we'll promote you so you can speak. And thanks again for filling out the poll at the beginning and the end. And I'm going to go ahead and hand it over to our Science Deputy Executive Officer, Henry DeBay.
Thank you, Dylan and Eva and Megan, and our public participation team who you might not be able to see but who's making all of this happen behind the scenes. So thank you. Good morning, everybody. My name is Henry DeBay. I'm the Deputy Executive Officer for Science at the Delta Stewardship Council, as Dylan mentioned. I'm also thrilled to welcome you all to today's seminar highlighting results from two projects funded by the Council as part of the 2021 Delta Research Awards. As Dylan has already said, but if you're just joining us and haven't already, please introduce yourself and share your name and affiliation in the chat, and don't be shy about using the Q&A and chat throughout the hour. So first, I did want to provide a bit of background about the Council and the Delta Science Program, which is the slide that you're looking at here. So the Delta Stewardship Council, including the Delta Science Program, was created by the California Legislature through the 2009 Delta Reform Act. And the Delta Science Program was charged with providing best possible scientific information to inform water and environmental decision-making in the Delta. And one of the ways that we provide that best possible science is by funding scientific research in the Delta, including through competitive research calls like the Delta Research Awards. And it's worth highlighting that our science funding efforts work hand in hand with two key tools that are pictured here, the Science Action Agenda and the Delta Science Tracker. So the Science Action Agenda is a collaboratively developed document that identifies management relevant research priorities. And the Delta Science Tracker is a web-based tool that inventories research projects across the Delta to promote communication and collaboration. And of course, the purpose of today and these seminars is to go beyond simply funding science, but ensuring that that science, including results and management implications, is communicated broadly. And next slide, please. So as Dylan mentioned, this is the third seminar of this series. Each seminar features two presentations on a shared theme. Today's seminar theme is multi-benefit wetland habitat and Delta food webs. And I want to take a moment to highlight the other forthcoming seminars. So next week's seminar will focus on collective action and co-production for integrated social ecological systems. Following that, we'll have seminars on HABs on June 26th, migratory fishes on July 10th, and we'll revisit the theme of multi-benefit wetland habitat and Delta food webs on July 17th, and have more to come after that. So please consider joining us for those upcoming seminars and spreading the word. Next slide. So I promise we're getting to what you're all here for momentarily, but lastly, I do want to point out some other Delta Science Program efforts you may be interested in or know folks who are. First, notices of intent for our 2025 Delta Science Fellowships are due on June 24th. These fellowships are competitively awarded to early career scientists at the master's, PhD, or postdoctoral levels to conduct research in the Delta. Second, we're in the early stages of facilitating an update to the Delta Science Plan. The Delta Science Plan is a collaboratively developed document and a companion to the Science Action Agenda that aims to improve the doing of science in the system. And to that end, the Delta Science Program has developed and circulated an essay as a means to socializing a proposed approach for the next Delta Science Plan update. And we'd love your feedback. Initial feedback will be reviewed starting July 12th. Third, the Council is organizing a Science for Communities event on July 12th. This event will showcase projects created through partnerships between Delta community-based organizations and scientists addressing topics ranging from air quality, harmful algal blooms, homelessness, and the human right to water. The event is in Stockton and free and open to the public. So please join us and please join us in spreading the word. Next slide. So again, today's seminar theme is multi-benefit wetland habitat and Delta food webs. As many of you know, wetlands are iconic and multifaceted ecosystems, and to assess and manage these special environments in the Delta, scientists are exploring them from every angle, from the sea level rise challenges that wetlands face to their contributions to the food web. So today's presentation, today's first presentation, is by Dr. Dennis Baldocchi. At UC Berkeley, Dr. Baldocchi's team has been measuring carbon dioxide and methane fluxes in and out of the wetlands in the Delta to assess carbon sequestration across a network of tidal and non-tidal wetlands, differing in age and salinity. They will also create maps of the Bay Delta with this information using remote sensing products. So this research addresses Science Actions 1, 2, and 3 from the 2017 to 2021 Science Action Agenda. And our second presentation, which we'll get to later, is by Dr. Peter Hernes and colleagues from UC Davis and USGS, where they looked at the quality and food web value of algal and detrital particles in a changing Bay Delta, including through the use of optical sensors. And this research addresses Science Actions 3, 4, and 5 from the 2021 Science Action Agenda. And each project's key research questions, findings, and management relevance will be summarized in an information sheet and available soon on our website. So with that, I would like to hand it over to Dr. Baldocchi. Thank you.
Okay. Well, good morning, everybody. It's a pleasure to be here. And talking with you all. My team is a group of biometeorologists, biogeochemists, ecosystem ecologists, trying to understand the fluxes of carbon dioxide in and out of these wetlands in order to figure out rates of carbon that's being lost through drainage and rates of carbon that can be accreted through the restoration of wetlands. We've started working in the Delta around 2007, and so I've had a huge team over the years of various graduate students, postdocs, and technicians. So I have their names listed here that kind of give you some sense. It does take a team to really do this work so well. Clearly, we're funded by the Delta Stewardship Council, but we're also part of what's called the AmeriFlux network, and so all of our data are available publicly on the AmeriFlux websites if people are interested. Next slide. I think many of you may have a sense of where we were and what we hope to get back to, but the Delta is really a unique ecosystem. It formed over several thousand years since the melting of the last glaciers. As you know, you could have walked out to the Farallones. The sea level was several hundred feet below today. Sacramento, San Joaquin rivers met this inland area, and as sea level rose slowly, wetland plants were able to grow, die, get buried underneath the water column because there's no oxygen for them to respire. And we built these peat levels, 10 meters thick or so. So very large stores of carbon. And if people are thinking about natural climate solutions, wetlands are really one of the better candidates because the flooding does inhibit the decomposition of organic matter, essentially. Next slide. But what happened is during the gold rush, our ancestors came to California. They went up to the Sierra Nevadas, started to mine gold. Many of them failed, and few of them remembered coming through the Delta. It was a tidal system. And so at low tide, you could actually see areas of exposed peat. Well, they're pretty smart. They realize that there's a high connection between the carbon content of peat, which can be 30–40%, and the nitrogen content. And if you're farming, and especially back in the late 1800s, early 20th century, you didn't have all the fertilizer around. And so drained wetlands could be, some of the most productive landscapes there are. And so there was a major efforts to drain the Delta. Next slide. And this legacy kind of came on two sets, both manual and technological. After the intercontinental trains was produced, you still had a lot of imported Chinese laborers, and many of them built some of the first levees around the Delta. Clearly, these were not very sustainable. And it was only really with the invention of the clamshell dredger by Dutra and the track layer by Benjamin Holt in Stockton that you could really start building levees and draining the Delta and actually farming them. So this led to this wholesale transformation of this landscape from this huge wetland with peat soil to now open drained land for agriculture. Next slide. And so you can look at these maps now, and the Delta has been this huge agricultural region now. Many people are growing corn, alfalfa, rice, pasture. But the biggest question is this sustainable? This is the reason we're here today. This is the reason we're being funded and doing this work. To help us understand our implications of wetland restoration, we also are making measurements on various landscapes. We have current measurements over alfalfa, corn, and we had some pasture and we had some rice in the past, essentially. Next. But the reality is this exposure of this peat soil allows the microbes to access oxygen and effectively decompose the organic matter. And so we've lost up to 10 meters of soil in many areas. I love this picture we took. You kind of see these boats flying from the Bay Area to Stockton on the San Joaquin River, essentially. It really gives you some sense of how much below land this is. And as you all know, this is the major conveyance pivot point for the water system of California. It's ringed by these weak, vulnerable levees. if these levees fail, and as Steve Deverall says, many islands have failed eventually, always, and they may do it so again. I've personally lived through seeing the floodings of Jones Track and Sherman Island and Brandon Island. And the reality is this is where our fresh water down the San Joaquin Valley in Los Angeles. And so a disruption of our water system in California could really have major implications on California's $4 trillion economy. So we got to do something about it. And we just can't keep business as usual. Next slide. So the reality is, let's restore wetlands. We've known anecdotally that they create large peat cores, and this is some recent data we took with Ariane Arias, and you can still see, and you can age these cores, and they are 4,000 years old. And this is one major aspect of natural climate solutions, is that you want to be able to restore ecosystems to a new state where the carbon is permanent. You hear a lot of work about, restoring grasslands and forests, but all that material gets decomposed and respired back to the atmosphere, or it's vulnerable to fire. Wetlands, for the most part, can circumvent that problem. So here we have the potential of landscapes that can be large carbon sinks with large rates of carbon uptake, and that the carbon that is produced in the soil will have long residence time. So it's definitely a win-win situation. I guess maybe the one limitation of solving the global problem through ecosystems is that the area of our land is limited. And I think the other point to stress is that with restoration projects, since the land is so far below sea level, we have to do it gradually. If you flood the whole island, you'll never revegetate it. And so this is part of the mindset of how we're doing this work. Next. So the reason to restore wetlands is that they can sequester carbon dioxide from the atmosphere, they can build peat, and these systems are wonderful because we have long growing seasons, much longer than many areas. We have plenty of sunlight, we have ample moisture to support the vegetation. We know that flooding inhibits respiration, and we also provide lots of other ecological services. With the other talk you'll see, we're doing food habitat, food webs. We're protecting the land from sea level rise, flood storms, bird habitat, etc. But the reality is, you can see from this picture, the landscapes can be complicated. They're very flat, but they have the different mix depending on the design of open water and vegetation. So, next. One of the trade-offs, though, is when you start flooding landscapes to retard respiration, you also remove oxygen, and that can lead to the productivity of archaea, another microbiological life form. And archaea are very, very adept of producing methane. And methane is a very strong greenhouse gas. It's maybe 30 times stronger than carbon dioxide. It doesn't have as long as a lifetime in the atmosphere. It might stay 15 years, whereas a molecule of carbon dioxide that's respired will stay maybe 300 years. So it's quite reactive. But the reality is that there is a scaling of increasing what's called gross primary productivity, or ecosystem photosynthesis will increase up to a rate of increasing methane. And part of the reason is that the plant roots have carbon exudates that are able to provide carbon compounds that the archaea like to eat, and they're able to decompose those and produce the methane, essentially. So if we're trying to understand rates of photosynthesis at the delta, trying to understand rates of carbon sequestration, we also have to measure methane production, and as well as water use also, because water is another major factor of this whole thing. Next. So what my team does differently than maybe other ecological scientists, and we're probably the first set of teams to do this in the history of the Delta, is that we're also trained as what's called micrometeorologists. I got my bachelor's degree at UC Davis in atmospheric sciences. So we can actually measure directly the flux of trace gases between the atmosphere and the landscape using this eddy covariance method that doesn't touch the sensors. It has a very large footprint, and it's a direct measurement. Ultimately, what we're doing is we're measuring vertical velocity every 10th of a second or so of the wind. And with these updrafts and downdrafts, we can actually measure the mixing ratio of the air in that, the CO2, the water vapor, the methane, and the temperature. And by looking at a covariance between fluctuations in vertical velocity from its mean and fluctuations in mixing ratio of the trace gases in their mean and averaging them over a half-hour period, that is by first principle the flux density. Our sensors are pretty robust, and we can kind of make these measurements quasi-continuously, day by day, hour by hour, year by year. As you'll see in a few moments, we have now produced up to about 100 site years of flux data in the Delta that can be used by you all as managers and decision makers on this, essentially. Next slide. So as scientists, we've tried to deploy these flux systems across different representative landscapes of the Delta. This is just a short picture of the ones that are being supported by this grant. So we have non-tidal wetlands that were restored on Twitchell and Sherman Island. The ones on Twitchell Island include West Pond, which was actually developed by USGS in 1997 as a small pilot project. And the east end was developed in 2013. And then Mayberry Slough is on Sherman Island, and that was broken ground in 2010. And we essentially put our flux towers right when the thing was done. And in more recent years, we are working with DWR and Gilbert Track, or Dutch Slough, which is literally across the street where I grew up. This is really a breached tidal wetland. It's freshwater. And with Ducks Unlimited and bond measures, we're supported at Hill Slough, which is near Sassoon Bay, which is mid in the estuary, brackish. That site is being naturally vegetated, so it still is mostly sediments. It's been slow. The Gilbert Track was actually planted at a large amount of money, but it's very, very, very productive. So again, it gives you different endpoints for decision makers, essentially on rates of wetland development, cost of the project, mix of vegetation and water. Next. So often, ecologists don't understand micrometeorologists, so we always get queries about how accurate our measurements are. And so we did some nice work with Ariane Arias Ortiz, my former postdoc. And by having 10 years of data, you can actually accumulate enough carbon in the sediments that you can kind of compare the methods. And we did this for both the Mayberry and the Twitchell Island sites, and you can see the comparisons that are within those statistical differences. We also were working with Patty Akawa, another former postdoc who's working at Cal State East Bay and has a site on the bay at Eden Landing, and there the methods don't work so well. And the reality is it's not that the micromet methods are wrong, it's just in tidal wetlands, there's just a lot of lateral flow of carbon. And so it just tells you that that's a so-called scale emergent property, that if you're measuring carbon fluxes in a closed wetland, the carbon budget's balanced. If you're doing it in a tidal wetland, there's a lot of lateral flow. We hope that carbon still stays in the estuary and goes out to the ocean shelves. We hope it's not being evacuated, but that is one of the major questions that people like us are still trying to figure out. And we're working with others like Adina Paytan at UC Santa Cruz on these issues. So, and Lisa has been an expert on this topic and done some very nice work at Rush Rants on this topic. So, next. So for today, I'll try and give you a little bit of results. Our key questions are: what are the net and gross carbon fluxes of non-tidal and tidal wetlands over time since restoration? How may these fluxes vary across the estuary? Because the reality is, as you go to more saline, that inhibits methane production, but also high salinity actually also inhibits plant productivity. So there is that trade-off up and down the estuary. What are the costs of methane to produce this carbon? And then finally, to try and think about predictions and scaling with models and remote sensing, what are the drivers of these fluxes and how can we predict them over time and space? Next. So I've been challenged to give you some takeaways, and one of the first takeaway is there's really no simple answer. I always like to kind of use an analogy of the quote from Anna Karenina, who says, all happy families are alike. Each unhappy family is unhappy in its own way. But we're kind of finding that with the Delta. Every field site kind of has its own separate story based on its location, its management projects, time since development, etc. And we can show this in the next figure here. So this is a time series of the data we've been collecting from various sites. I'll point your eye to the y-axis, which is in terms of grams per carbon per meter squared per year. The plus values are actually a source of the ecosystem and a gain of carbon by the atmosphere, and the negative values are the sink of carbon from the atmosphere and accretion of carbon. So the ideal is to have projects that are below the zero line and are sinks of carbon. Well, you see, first of all, it's just, the corn is just off the charts. It's just burning through the delta. And these are on Bolden Island. In the past, we were seeing maybe 1,000 grams of carbon per meter squared per year. In the last couple of years, we've actually pushing past 1,500. Remember, corn, even though it's a C4 plant, it isn't annual. It's fallow much of the year. And so the soils can be exposed to oxygen and just decompose. The only good thing is when you have flooding in the winter for ducks, that can kind of inhibit some of the respiration. Alfalfa can be a modest sink. It's a perennial, has a long growing season, goes through many cuts. But the reality is, where is that product going? Well, it's being used to feed cows, and the cows respire that, and ultimately that carbon will retain back in the atmosphere. The wetlands now are interesting because they are good sinks. However, in the early years, since you're disturbing the soil so much and they've been drained, they can be a carbon source until they are vegetated and then starting to acquire carbon. And they take up on the order of 400 to 800 grams per meter squared. I like to give you all a perspective. I can take a sheet of a computer paper and weigh it and scale it out to the size of a meter, and that weighs about 75 grams essentially. So at the best, our ecosystem can take up the weight of 10 pieces of computer paper, which is pretty good, nice part of a ream essentially. So this gives you some sense of the variety of conditions we have essentially. Next slide. So in summation, corn's a huge carbon source. Perennial alfalfa and flooded rice are moderate sink, but their carbon's consumed off-site and returned to the atmosphere. Non-tidal wetlands, naturally revegetated, are initially carbon sources, then become good carbon sinks. Freshwater, revegetated, tidal wetlands are among the strongest carbon sinks in the world. Flooding inhibits respiration and buries carbon for climatically long times, but there is a cost of carbon sequestration in terms of large methane fluxes, and with water, we actually can use up to 1 meter of water, close to potential evaporation in California, where water is quite precious. Next. So this is a histogram. I've been a principal investigator of this global FlexNet project for over 20 years or so. And so I've been compiling data from all of my colleagues from many terrestrial ecosystems. So this is a histogram essentially. And the x-axis gives you some sense of the magnitude of this net ecosystem exchange. And the Dutch Sluice site in the Delta is one of the top 1 percentile of carbon flux uptake in the world. You look at these iconic redwood forests and think maybe they're perennial, huge, and they're big carbon sinks. But our little humble Oakley Dutch Sluice is among one of the largest carbon sinks in the world. And again, to give you some sense of mass, it's about the weight of a bottle of wine. Bottle of wine, 750 milliliters, 750 grams, spread over a meter. Our other ecosystems are more like the mass of a can of beer, essentially, where they're taking up 350 to 400 or so. So it gives you some sense of where we are, and really encouragement that these restoration projects are important because these are really novel and very effective ecosystems. Next. This kind of, again, gets back to the points I've made earlier, but with real data. So the point is, if you have a flooded ecosystem, you can look at the relationship of assimilation, photosynthesis, which is the gross primary productivity on the x-axis, versus ecosystem respiration. They always scale, but the cool thing is by flooding, that ratio is about 40%. So essentially, we lose about 40% of our assimilation through respiration. There's always that cost. But realize if I look at the right panel, that's the Ameriflux data for terrestrial ecosystems, that slope is about 0.85 or 85%. So the difference between photosynthesis and respiration is much, much tinier for upland ecosystems and wetlands. So this really kind of makes that point, essentially, how effective the practice of flooding wetlands is in terms of sequestering carbon. Next. Oh, and just one final thing is, go back. It's this type of relationships that we hope we can use to upscale these data from the expensive flux towers to the region with remote sensing. We can measure quite well reflected sunlight of different colors to actually estimate this GPP factor. If we know this slope for between respiration and GPP, we can actually map net ecosystem exchange across the landscape in a very cost-effective way, especially with new satellites like Planet Labs that can essentially give us information on 3-meter resolution. Okay, next. But as I mentioned, there is this biogeochemical compromise. The more you sequester carbon, the higher you'll have methane rates. And so these are data from my former grad student, Kyle Hernes, but it just kind of makes this point, essentially. So these wetlands can be some of the most prodigious methane producers in the world, mostly because we have these high rates of photosynthesis, and therefore the methane's tied to it. So yeah, next. What's really interesting now is starting to be able to compare the tidal versus non-tidal wetlands. And so this is a nice time series of daily carbon fluxes between Dutch Slough or Gilbert Track at near Oakley and the East End thing. And what's really important is that we have a much longer growing season, even though these are 10 kilometers away. What happens with the non-tidal system is a lot of legacy dead biomass builds up and builds up and builds up. And it starts blocking the growth of the new vegetation, number one. So your photons are intercepting dead tules and new tules. And it's also inhibiting the sun from warming the water. And so it delays the leaf out of the wetlands. So effectively, these older non-tidal wetlands actually are delayed in terms of the green up by up to a month. And so you have a much shorter growing season to take up carbon, essentially. The methane of both ecosystems are large and comparable, essentially, but you definitely see this big difference between tidal and non-tidal. So the non-tidal system may take up about 400 grams of carbon, and the tidal system will take up about 800 grams of carbon per meter squared per year. So, next. The other issue is what's happening at a given site. I really thank everyone for having the wisdom to fund us for a long time, because if we had just made 3 years of measurements and walked away, we'd come up with a completely different conclusion. This is our data from Mayberry, which is kind of at the confluence of the Sacramento and San Joaquin River. It's been a good carbon sink for many years, but we've had some droughts that have limited the flow of water. We've had some insect infestation, and so we've had a few years actually with poor carbon uptake. In the earliest years, we had humongous amounts of methane emitted from these systems. As you can see in the lower panel, up to 63 grams of carbon equivalent from methane. And that's dropped by 1/3 in recent years. And so we're still trying to figure out the what's, how's, and why's in terms of how this is relating to our salinity and the changing footprint. Yeah, I'm almost done. So next slide. So one of the challenges with us all was to try to create some mathematical models and statistical models to try to upscale this. And so we've been using this method called alternating conditionals expectations to try to figure out what the dominant drivers are, essentially. And so this kind of gives you some sense of how well it's working. So next slide. And so these are the overarching messages to take home. I guess if you're a farmer, realize that business as usual may not be sustainable as the delta continues to degrade. I know we're trying to have farm with rice as a way to do this, but you know, I'm thinking 50 to 100 years down the road. We have to protect our delta because it is the conveyance system for a $4 trillion economy. Restoration projects can be very effective. They're huge carbon sinks, but it is a matter of scale. You know, we're talking about what, I forget, 700,000 acres of the delta that we need to restore. One size doesn't fit all. There's various ways to design these projects essentially. There's definitely no free lunch, and we have to be careful about not throwing the baby out with the bathwater. Just because I say there's methane, it's not to say let's not restore wetlands also. So I think maybe there's one more final slide just to final up. Oh yeah, future studies. So I can stop now if you all want, and we can have the discussion that you'd like. Thank you for your attention. It's been a pleasure.
Thank you so much, Dr. Baldocchi, to you and your team for all of this work and for this talk. So I'm just scanning for some questions here. Maybe to just kick things off, especially for folks who aren't as steeped in this kind of work, if you could talk a little bit about the different kinds of fluxes, you know, lateral versus other kinds, and the ways that you go about measuring the carbon fluxes.
Right. So we've talked mostly about the vertical fluxes. At the Dutch slough, we're also trying to look at the flows of carbon going in and out of this wetland. And so we actually have a velocity sensor that's measuring the tidal flows in and out. And then we have measurements of carbon in the water column. And so we can try to integrate what those carbon fluxes are. What's nice about the Dutch slough is that there's just one opening. And so we can actually kind of look at, well, we treat this as a bathtub, your bathtub interval. And so if you know the flows going in, the flows going out, the velocities and the concentration, that you can kind of calculate those. It's a little harder to do because the vegetation fouls things. These things aren't as reliable as the other ones, but we're trying to get some good measurements for that. So.
Thank you very much. Okay, we've got some questions in the Q&A. Let's see, I've got, I'm going to start off with Dr. Lisa Marie Wyndham Myers' question, which is, with some potential future land cover types coming in, both intentional, like floating peatlands, and unintentional invasive plants like Phragmites or aquatic vegetation, do you think we can predict their carbon benefits as being similar to the more historic Thule freshwater wetlands? And then there's another question. So that's the first one.
I don't have the data on those. I do know Steve Deverell and colleagues have been actually making some nice measurements of the floating peatlands, and they do have some promise essentially. So I guess the biggest question is forming those peatlands, because if you have to, what they did, I understand, is they cut out peat from one of our other wetlands and moved it and put it out into the river systems and floated it. So there you have to be careful about the net exchange essentially. So the Phragmites, I'm not sure. I'm sure there's data from Egypt and other locations of the world. And be honest with you, these systems are really efficient. They tend to drive photosynthesis at about 2% of the absorbed energy. They form closed canopies. So in some respects, as a biometeorologist, from a first principles perspective, I would think that rates of carbon uptake could be similar. It gets back to really what the respiration is from those landscapes and how well they're flooded or not. I mean, the big take home, I think, in terms of trying to manage the delta, is I really favor a perennial landscape vegetation as opposed to the annuals. A lot of the annuals, are fallow so much of the year, and they're all ultimately solar collectors and trying to convert that solar energy into stored carbon compounds. And so if you have a full canopy with a high leaf area index, you're absorbing 90-95% of the sunlight. You're converting that at 2% efficiency for 200 plus days, essentially. And that's probably the best you can do until the first frosts come and things slow down.
Great, thank you so much. Lisa Marie's second question is, do you suspect the biggest climate drivers to pay attention to for predicting climate mitigation benefits are temperature or salinity, or something else?
Oh, I mean, the delta gets exposed to these really high temperatures. And so even though they have ample water and they're flooded, you get some really high vapor pressure deficits that still, I think, can cause some physiological stress. I mean, the reality is these things have these huge orecchima, so they can have oxygen go down to the roots to keep the roots alive. And so I think on a hot day, they kind of shut down a little bit. So, salinity is an interesting one. I mean, these plants have been exposed to salinity, historically before the dams and after the dams. And so they're just big gradients on that. And I guess you just have to figure out each location of what that salinity level is. Yeah.
Great. Thank you. Okay, we've got some other questions here. One from Dylan Chappell, who I think a program manager of the Adaptive Management Unit at the Council. His question is, if you could collect one additional variable to your current data set, what would it be, why, and what additional questions would it allow you to answer?
Well, we just bought another sensor to do a better job in our lateral lateral flows, essentially. So the one we were using was crapping out. So again, for these tidal systems, we want to just do a better job at measuring the lateral flows of carbon in and out of these wetlands. So that's what we're working on at this stage. So.
Great.
Been really happy, to be honest with you, with our eddy covariance sensors. I mean, these things we bought with DWR money back in 2010 or so, and they're still running. This is 2014. So we had to repair a few of them and stuff, but these work on solar panels. And so we're actually being able to go off into the wetlands and study the methane and carbon where it's actually happening. When I first started this work in 2007, we had to use closed path sensors that use scroll pumps, and we had to actually make our measurements close to a power line. And that really limited the ability to do the work. So the idea of having open path sensors running on solar panels has just revolutionized this game, essentially.
Very cool. Yeah, those towers are pretty prolific in generating data. Okay, we've got a question from senior environmental scientist Shoko Shimbrat. Curious what the time scale is when you say wetlands can store as much as 10 sheets of paper or as much carbon as a bottle of wine. Over what period of time are we talking about?
Yeah, that was the yearly integration. So each year they can build that much carbon, essentially. And I think you look at the data from West Pond and Steve Deverell and others. I mean, I think we build a couple inches of carbon or soil per year. The reality is converting that into stored carbon, because in early years you have what's called proto-peat. It's not well packed, and so it takes many years for the actual peat to form. And so it's much easier actually to make the measurements of direct carbon fluxes with our eddy covariance, because it does take a decade or plus, as I already showed, to actually take soil cores. It's cheaper, it's manual, but the rates of carbon accretion are such slow to make significant measurements of differences. It takes time to integrate.
Great, thank you. And thank you also to the folks who are populating the Q&A. And if you can't see it, it's because it needs to be answered first, and then it gets moved over to the answered column. So they are popping up in kind of a delay. There's an anonymous attendee, sorry, I can't see who this is, that's asking, regarding Dutch Slough, can you clarify the location of data collection efforts? What kind of wetlands it contains, ponds, ? Vegetated ? And what ongoing data collection efforts you are aware of?
Yeah, well, it was a low tidal marsh, essentially. So it was originally the Burroughs Dairy from the early 1900s, essentially. And what the state did was they kind of moved some dirt around and then breached the levee. And so water was able to come in a tidal sense there. And then they revegetated it with, I guess, tules and cattails, essentially. So there are various levels. They tried to create kind of the natural dendritic structure of these historic wetlands, essentially. They have channels here and there. And so when I have a flex tower, depending on the wind direction, I'll see a different fraction of open water and vegetation, essentially. So that does change the measurement I make from different wind directions, essentially. So we do have to consider that. And so my student Robert Short and us have collected LiDAR data. So we have now really detailed three-dimensional maps of these landscapes with LiDAR. We flew drones with a consulting company that Dylan supported both during the winter time when the vegetation was dead and dormant, and then during the green system. So we can actually map this very, very well, essentially. And again, this data can be shared with everybody to use.
Great, thank you so much. Okay, we've got about 5 more minutes for questions, and of course the questions are really coming in now. So we'll try to squeeze in as many as possible. The next one is from Senior Environmental Scientist Denise Colombano at the Council. Her question is, what are you learning about the new Hill Slough restoration and how it compares to what is known about the historic marsh in Rush Ranch?
I guess we just have to be patient. We're seeing the tules expand and grow to the north of us, but it tends to be outside our flux footprint. We're starting to see a few tules popping up. Aaron Will, the manager, gave me a good comment. The winds come from the Carquinez Strait, and there's really very few wetlands to that direction. So there's very little or reproductive material blowing into our system. And so it's just take a while for it to expand. Now, we had the same thing with Mayberry, that it wasn't vegetated, and within 2 or 3 years, it was full canopy. And I think the reality with Hill Slough is in 4 or 5 years, you probably won't know the difference. But we are working in this first 2 or 3 year period, and it still is mostly open, essentially. So I think for the managers, just be patient, I think, and be supportive, and just it takes time in different locations. So that's really my takeaway from that. It's sad to say. I wish it was all vegetated myself.
I feel like being patient is a good rule for most science.
Patient and persistence. I've learned, embrace serendipity, be patient, and be persistent. Those are probably the 3 things as a senior scientist I've learned in this.
Good.
And again, this Delta work, I had a field trip. I'd heard about the Dutch slough in 2002 and brought a bunch of students as a field trip to see it. And it was a dream in the eye. And it really wasn't until 20 years later that land had been broken. And I was able to kind of do work there. So again, patience and persistence. And a lot of investment from a lot of managers in terms of permits, funding, engineering. I mean, it really does take a village here.
Okay, I think we have time for just a couple more questions. We got one from Norman Johns at the State Water Resources Control Board. Norman's question is, in the historical cores of peat soils, can you describe how much, if any, mineral, i.e. non-organic content was within those?
Ooh, I don't know the exact number. Julie Drexler really is the expert, and I mostly read her papers. And Lisa Marie, I think, is a co-author on some of these papers. But the thing I guess I've learned anecdotally is that the Delta isn't all peat. This is the lesson we learned when we first worked on our rice. We actually started looking at Google Earth, and you could actually see mineral deposits from the channels. So historically, you would have had runoff from the Sierras and weathering, and so it would have had a mix of peat and mineral soil. And I had an undergraduate recently interested in native burns. I showed that first picture with the big tules and that big fire. That was kind of inspiring to us. But the Native Americans, because they're all that duff that would build up, it was difficult to hunt. It was difficult to have really fresh tules to make their baskets. So they burned the Delta periodically to refresh its growth. And we actually did some soil cores, and we actually see some charcoal levels. And I think we were able to date some of those to the late 1700s or so. And we started talking to Julie, and she went back and looked at a bunch of her cores, and she also found charcoal at various dates. And so, these are some interesting questions that need to be resolved into the future. But it's great question. And to be honest with you, the corn is on the peat with the highest nitrogen, and the alfalfa tends to be more along the edges of the rivers, whereas there's more mineral soils. And I learned when I was at Davis, all your pear orchards and stuff actually are along the river edges also, because that's where the mineral soils would have run off over floods and actually built, again, thicker, less peaty, saturated soils that are good for the pear orchards and stuff. So the Delta is very mixed with peat and mineral soil. Thanks for the question. Good.
Thank you so much. So I'm actually being told we need to wrap it up at this point, but Dr. Baldocchi, thank you again for your talk. If you can stick around, I think we're going to add the outstanding questions to the chat, if you wouldn't mind answering those there. And then one of the questions that we had is if you're okay sharing your slides. We'd love to make those available. Great. Thank you so much.
I hope I was able to address the questions for the general people that they wanted here. And so, yeah. So thanks a million.
Thank you very much. Okay, moving on to our next presenter. I'll ask for the next slide, please. Great. So again, I just want to introduce Dr. Peter Hernes and colleagues from UC Davis and USGS. They have looked at the quality and food web value of algal and detrital particles in the changing Bay Delta, including through the use of optical sensors. You can see here that this work advances science actions 3, 4, and 5 from the 2021 Science Action Agenda. So again, big thanks to you, Dr. Hernes, for being here, and over to you.
All right, thanks for the invitation. I'm not sure who is all here with me right now. I know that Brian had a conflict. I don't know if Tamara has been able to get on board here or not, but hopefully if there's any questions specific for USGS, one of them will be around to be able to address those. This is a joint project between UC Davis. We got our funding through the Delta Stewardship Council, and USGS got their portion from the state water contractors. So we've been working together. And then Madison Mathers is my current graduate student, and she's been doing all the heavy lifting in my group. And a lot of the work that's being, or the figures that are being presented today are ones that she has been putting together through her dissertation. Let's see. Okay, you're reading my mind. I like that on the slide advancement. So I work with carbon and water. Brian and I actually went to the same graduate group at University of Washington way back a long time ago. How we ended up in the same location working together seems a bit of a mystery. I think that's the serendipity that Dennis was talking about. But we have a carbon focus, and one of the things that we've been, when we look at the research that's been going on in the Delta, there's been kind of a dearth of carbon research. And one of the reasons for that has been that there's been so much emphasis on phytoplankton as the primary base of the food web. And when you do the mass balance on it, the phytoplankton alone isn't enough. And so the question has been, what else is out there? What else is supporting the food web? And so historically, you look at the slide on the left here, you can see that chlorophyll A and primary production have been declining over the years, and that corresponds with this decline in fish. And so obviously, if the food supply is declining, there's a problem with the fish. And so one of the original justifications for this particular project was that USGS has this monitoring boat, where they do a lot of underway monitoring, and the idea was to look at the nutrient field after the upgrade to the wastewater treatment plant and see how that impacts food web, phytoplankton, things like that. So even we are not immune to sort of looking at this idea that phytoplankton are the most important part of the food web, and they probably are, but it's not the only thing. And so one of the things that we've been doing here at UC Davis has been focusing more on the other potential sources of carbon to the lower food web. And particularly, we are looking at detrital material, and that we think mostly is coming from wetlands. And so that's sort of the hook that we have into kind of the work that Dennis is doing, is that we are hypothesizing, I guess, that as you start to rebuild wetlands, you're going to start to increase the amount of detrital particles in the delta. And hopefully that will have a benefit for the lower food web. Next slide. So goals, in the original proposal was to do these wide area mapping surveys. And so, USGS has got a boat that's fitted with a lot of in situ underway monitoring so that they can get things along the way. But then we also stop at 30 to 35 stations on each one of these things to collect discrete samples. Those discrete samples would come, a lot of it would come through our lab. Maddie was busy filtering. I would chip in and filter. So, we'd have this big rush of 30 samples coming in that had a lot of filtering to do. And then we would do analysis on those, and we'll talk a little bit about that in a bit. So part of what we're trying to do then in sort of build up our knowledge or build up our understanding of POC cycling dynamics is to be able to compare this to data in the past. And there hasn't been a lot. I published a paper from data from 10 years ago. Before that, there was data from 20 years ago. There just really hasn't been a lot that's been happening with POC research. And then in our lab, we've been using biomarkers and amplicon metagenomics to try to look at the sources of these particles to find out if they really are coming, the detrital material is coming from wetland. But what's beautiful about the amplicon metagenomics is that you get species level. So you don't just learn that your particles are coming from wetlands, you learn exactly what species in the wetlands they are coming from. And it's not quantitative, but it gives you unambiguous information about where these particles are coming from. And then the 3 action areas are listed at the bottom. Those have been referenced before. I'm not going to talk specifically about those today unless I've got some questions at the end. Mostly I'm focused on the science. Next slide. So this is some research that came from a former student of mine, Jennifer Harfman, who had a Delta Science Fellowship and did this work as part of her Delta Science Fellowship. She used the amplicon metagenomics in a series of feeding experiments. And that's on the left, that upper pie graph there shows you with amplicon metagenomics what the sources of particulates were in the water column. And you can see in there that less than 1% of it was coming from vascular plants. The rest was coming from phytoplankton, from things in the water. When she fed that water sample to copepods in a feeding experiment and looked at what was in their guts, it was amazing. Almost 2/3 of what they ingested was coming from vascular plants, that they were selectively picking out this detrital material. It wasn't just that it was passively going in, they were actually selectively. So this is kind of one of those reasons where, or one of those times where, you know, the non-quantitative nature of metagenomics didn't really matter. It's just direct evidence that they're actually, you know, that they're doing this, that they're selective feeders. Then she did another set of experiments there. Look on the right. If you look at the red line there, that one is the same copepods feeding on just chlorella algae. If you look at the kind of the pinkish line there, it's pure tule. So feeding on tule, their survival in pure tule isn't very good. They don't last for very long on just tule alone. But when you combine them together, the tule plus the chlorella, you get enhanced survival. So again, another line of evidence here that detrital material is an important part of the lower food web and that we need to pay more attention to it. Next slide. So when we were out there for these survey cruises was 21, 22, and you can kind of see that in relation to the years around. Of course, you know that we've been here in California kind of over this extended drought since 2012 with some rainy periods here and there as well. But we hit a couple of very dry years in 21, 22. Next slide. And they're historically low particle years. So this is a plot that Maddie put together with TSS at the Sacramento River Freeport gauging stations. The 2 years that we sampled are in that pinkish purple on the far right. And you can see that it's sort of the particles are lower than anything else that we've had. So in some ways, I suppose it's, you know, what we were measuring is an example of the extreme. But with climate change, it's hard to say. In climate change, we could have extended and severe droughts. So this may be a condition that we have to get used to here, this sort of low particle thing. And I think it's also kind of important to recognize, and Dennis highlighted this as well with his wonderful historical perspective on the Delta and how the Delta's changed and all the crises around that. And I usually lead with a lot of that stuff too. And so I'm kind of happy that he already hits you up with that so that I don't have to spend as much time talking about all the issues with the Delta. But you know, the Delta has been a very disturbed system for, you know, in a lot of different ways. There was the hydraulic mining, there's erosion from, you know, landscape changes in agriculture and things like that. In some ways, you know, this decline here probably reflects just sort of the natural processes. Farming's been getting a lot better about controlling erosion. There's a lot more stability as landscapes start to get higher vegetation rates on them, things like that too. So, I mean, I think it was inevitable that the delta, the Sacramento River, was going to decrease in particles just due to these different kinds of pressures and things like that. I don't know if anybody predicted that it would get this low, but here we are, some pretty low particles. Next slide. So the different types of tools that we've been using there, the boat high resolution mapping with sensors and water samples, water flow that's kind of going in continuous flow path sensors and things like that. And it allows the ability to do these underway maps. I think the one that's showed here is probably a nutrient map, a nutrient field, but we're not really talking about that one particular today. Amplicon metagenomics, just a flow chart of what that looks like, where you're extracting DNA out of your samples and you're comparing it to primers that allow you to get species level identification. And then another tool that we use in my lab is lignin biomarkers. That's the chicken wire structure off on the right. And that is it's part of vascular plants. The cellulose is what gives the plant structure, allows it to grow up and have rigidity and form. The lignin is what holds that cellulose together. It's like the glue. It's a random polymer, which means that it doesn't degrade very quickly. It's not lock and key. When you put it into anoxic environments, it degrades very, very slowly. And so, part of my curiosity in listening to Dennis's talk is just that, when you put lignin in those anoxic environments, it sticks around. So we have some data for that as well. In the early periods of wetlands, lignin degrades quite quickly in the sediments because you haven't established those anoxic conditions. But once you hit anoxia, lignin sticks around a lot. It's one of those oxygen sensitive materials that would help build accretion when you've got the anoxic conditions. In our case, we use it as a source biomarker. Syringeal phenols are only produced by angiosperms. Synamal phenols are only produced by soft tissues. And then we use comparisons of acid aldehyde ratios to kind of look at diagenetic trends as well. Next slide. So this gives you an idea of just, a couple of snapshots here, May compared to October in both 21 and 22. And so you can see when you've got a lot of high energy in May with the spring freshet, the spring flows and things like that. It's probably bringing in more sediment into the delta, but it's also got the capacity for stirring more sediment up from the bed sediment. So this complication of looking at sediments in the delta is that it can be tricky sometimes to understand if it's being brought in or whether if it's just resuspension. So anytime you get a big storm, big winds, things like that with enough fetch, you're going to stir things up, especially in Grizzly Bay, Sassoon Marsh, that area. So in October, when the flows are low there's not nearly as much particle action happening in the delta. Next slide. So this is one of the ways that Maddie has been using metagenomics. She has, is able to take all the phytoplankton data and organize it by type. And of course, different phytoplankton have different food value. One of the things that's sort of critical towards zooplankton are these long chain essential fatty acids, which is one of the reasons why they can't grow on tule. Tule doesn't have that. But tule does have carbon. And so if you have a phytoplankton that's producing enough of these long chain essential fatty acids, then you can use detrital wetland material to augment the carbon portion of it. So the higher food value phytoplankton are cryptophytes, diatoms, dinoflagellates. And so Maddie sort of built this map. This is a composite of all 6 sampling times all at once. And you can kind of see that, some of the higher value food is sort of concentrated in the northern delta, on the Sac River thing. Out on the, you can kind of see a lower food web value sort of in the southeast, and that one out there in Grizzly Bay, which I'm not quite sure what these. Yeah, I don't know how much more I can say about that one. That might be a question for Maddie or for other people who are more versed in phytoplankton than I am. Next slide. And then we can also do the same thing for vascular plants through this metagenomics. And one of the kind of fun parts of this study was finding things like sweet potatoes, yam DNA in these samples. And you wonder, like, well, where does the yam DNA come from? And then you start looking at a map of the delta and you realize that, well, they're growing yams in the delta. And so, you know, some direct evidence here that the crops that are being grown in the delta, even though they're kind of internal, you know, they're not directly flushing out to the delta other than through the I guess through the tile drains, you know, that you have to drain them to keep the root zone aerated. There must be enough DNA that's coming out of those pumps to be able to influence the particles in the delta. And so in a lot of these cases, at least in terms of the DNA, more than half the DNA was coming from these agricultural plants that are grown within the delta. And then that light blue, that's sort of wetland material, aquatic macrophytes, aquatic vascular plants. And of course, out there in Grizzly Bay, you see this a big spike of that, which makes sense because there's a lot of wetland out in that area. And then the terrestrial material in the light green, that is mostly stuff that we have. I guess we're mostly lumping that into the bin of stuff that comes in on the rivers, that flows in from the Sacramento River, from the San Joaquin River, so it's imported. So the agriculture and aquatic would be sort of internally sourced, the terrestrial would be externally sourced. Next slide. Just a quick look at, you know, how we use lignin biomarkers then. Because you do have information about sources, we do these property plots. So on the y-axis is S to V, syringal to vanilla phenols. On the x-axis is C to V, synamyl to vanilla phenols. And that tends to be kind of a marker in broad terms about different sources of material. So in black, you can see the samples, the discrete samples that were collected and measured during the mapping surveys, and the X's, the colored X's, are different aquatic macrophytes, aquatic, you know, submerged vegetation, emergent vegetation, things like that. And so you can see that in terms of lignin sourcing here, the particles that we are getting from the water column don't match up all that well with the particles that are coming that we measured, or the plant materials that we measured from these wetland plant types. There are different ways that you can interpret that. One of the ways that you can interpret that is that that the material that's coming off these wetlands gets chewed on, gets taken up by zooplankton, gets you know, I guess I'm trying to think of what the right word for it is anymore. Fecal pellets, things like that, that it gets processed. You remove some of the value, you change the lignin composition, and then it comes out looking more like the black circles. Another idea could be, of course, that we're just looking at the wrong stuff, that there's, you know, that the things that we were measuring in terms of plant sources doesn't exactly match up with what's in the particles themselves. But, you know, the data that we have, at least in terms of metagenomics, and this is where lignin and metagenomics can kind of play really well together, because one is quantitative, one is qualitative. So you get some targeted information based on, so they I guess they cover the strengths and weaknesses of each other. But my own interpretation, my own sense of what this works, how this works, is that what you're seeing here is just a reflection of processing. That these fresh materials that you're seeing with these colored X's gets processed down, it's getting worked, and then it ends up being kind of falling more into the black dot buckets. Next slide. So we can also use our biomarkers to do more of a quantitative perspective on where the sources of POC are in the delta. And so this is one of the things. So you use the chlorophyll, which that looks like I've spelled incorrectly, so that's kind of embarrassing. The chlorophyll, there's a scaling factor that's been used in the delta where you multiply the chlorophyll times, I think, about 35 in order to get phytoplankton biomass. That's how we derive that phytoplankton term in green. And then we use lignin biomarker, carbon normalized yields of lignin, to try to distinguish between the aquatic macrophytes, the aquatic vascular plant materials, and then the terrestrial things that are coming in. And so the take home here is that there's a lot of stuff that's being produced within the delta. In most cases here, more than half the carbon that we were seeing in the particulate field was coming from within the delta. And so in some ways, that means that the management, I mean, it's good news in the sense that we can control through management what this particle field looks like. And of course, wetland restoration is one of the tools that we have in the bucket to be able to do that. I mean, I've been kind of curious thinking about this wetland restoration and then in the interior of these submerged islands. How do you get that material out into the water column where it could be useful for fish? When it's at sea level there, you've got tidal pumping and tidal flushing that sort of brings this material out into the water column. When it's internal on the islands there, I wonder about how you could take advantage of that, how you could use this wetland restoration on the interior to try to augment the food supply out in the delta. But anyway, so this is the real take home here is that there's a lot of action that's going on in the delta. Next slide. This is looking at, I think, POC budgets, where the carbon's coming from and where it goes. And one of the things that we thought was rather surprising here was that up to 37% is lost via exports to the State Water Project, Central Valley Project, Contra Costa. Next slide. And then just an overall summary. Obviously, particles have been declining, which when it's the base of the food web, that's a problem. We see a lot of variation in spatial quality, which I guess gives us an idea about places that we may or may want to target in terms of trying to augment food supply, food quality. Phytoplankton varies across the delta, and it seems to be a lot more higher quality in areas that have a lot of energy in them. Terrestrial material comprises a significant fraction of the organic matter in particles, which of course is one of the themes here that we want to know whether that detrital material can end up in the food web. The aquatic vascular plants also make up a huge fraction, and of course, we think that that's a good thing, that can help augment the fish. And then the last point, just re-emphasizing here that the water exports again are taking particles out of the delta, which is probably not a good thing. All right, thanks.
Oh, I was on mute. Thank you so much, Dr. Hernes, for that talk. Really appreciate it. So we've got some questions already. The first one is from Dylan Chappell. With regards to detrital POC sourcing, are tidal wetland plants like Thule considered terrestrial or aquatic?
We've been putting those in the aquatic bucket. Yeah. And we see a lot of Thule DNA out there.
Great. Second question from Dr. Lisa Marie Wyndham Myers. Sorry, I need to answer that question. Okay. Wow. Even in these drier years of 2021 and 2022, those are high POC concentrations, 2 milligrams per liter in some places. Looks like in some places, those high values are from aquatic vegetation and sometimes from terrestrial sources. Would you expect they could go even higher in wet years?
I think this is where it kind of gets a little bit tricky, knowing whether this is like increased particle formation or whether it's resuspension off the beds. I think that's a really kind of a challenging question to tease apart at some point. But I mean, I think they have to be able to go higher than that. I mean, if you look at this historically low particle year, I mean, historical precedence tells me that they have been higher in the past, and so it makes me think that they could go higher again.
Great, thank you. A second question from Lisa Marie is, thinking about high value food, when you do metagenomics, can you also see the difference between algae sources, pelagic versus benthic, and also epiphytic algae that grows on the aquatic veg? It can be very dense.
You can if the DNA is there, you can see it. Yes. I don't know enough about the species to go back into the DNA and find out whether we have been getting benthic organisms or not. That would be a question I'd have to punt on and ask Maddie about and see if that's something that she could dig up. DNA as a biomarker, which is essentially how we're using it, is something that we still have to learn a little bit about, selective degradation, how long it lasts, what it means to, when you get phytoplankton in the water column that's alive, we kind of assume that that it may, well, we're guessing it might be overrepresented in terms of the total amount of DNA that we get in a sample compared to detrital material that's been, potentially senesced, things like that too. So there's some information that we still need to learn about how DNA works as a biomarker. I think I'm kind of talking around the question there, but if it's there, we can see it. So if those benthic phytoplankton are there, I think we should be able to see it.
Great, thank you. We've got another question from Dr. Shoko Shimbrat. I'm wondering if your labs or maybe collaborators have done any experiments to increase POC and lignin in the delta. For example, I was recently introduced to the German concept of Hugelkultur, a horticultural technique of building up decaying wood debris into mounds. Could that be appropriate and effective here in the delta?
I think the context that I've wondered about with things like that is for wetland accretion, for bringing in outside materials and burying them in the wetland as a way to try to accrete things faster. I don't know how that would work for increasing particles in the delta. I don't know if that's like a one-to-one response. If you brought it in and you put it in, would it end up in the delta? I mean, lignin itself has never been considered a high value food product. In fact, there's a lot of people who assume that it takes more energy to decompose lignin than you get back out of it. And so typically organisms will go after the lignin to get to the cellulose. So they'll put energy into that because it unlocks the candy underneath. So lignin is not something I would necessarily bring in for, it's not something I would bring in for particles and trying to prop up the food web, but it is something I would bring in for trying to get wetland accretion, because again, it's one of those oxygen sensitive materials, and when things go anoxic, you're going to get preservation.
Thank you. Another question we have is, and forgive me if you've already covered this to some extent, but recognizing the great work in progress made during this project, where do you anticipate future efforts focusing energy and resources?
Well, I think one of the things that I would like to do, I like to tie things into mass balance. I don't know that we have a really good sense, and Brian, you can maybe correct me on this because I see Brian's on right now. I don't really have a good sense yet about how much particles of these detrital particles are actually exported out of wetlands. Like, when you go through the ebb cycle of a tide and you're getting the waters coming back off of this thing. I don't know what's coming off. And I think we kind of need to know some more information just about the potential for a tidal wetland to eject particles out into the delta, so we can kind of get an idea about what you're going to get when you start doing wetland restoration in terms of scale. How much realistically can you augment particles within the delta? So I think, going after that quantitative aspect would be good. We have the information that we have in terms of these feeding experiments with copepods are based on just one or two types of zooplankton. And so, we've been wanting to be able to expand that to other types to see how more general this incorporation of detrital material is within the lower food web. There's been some methodology issues with that. We've been trying to answer that question, but we've been having some methodology problems with our field samples, in which the metagenomics just is giving us data that we don't quite believe yet. So we've got some technological hurdles to get around in terms of answering that question. So there's a couple ideas.
Great, thank you so much. Brian, I see you've been promoted to panelist. Just want to see if there's anything you wanted to add. I'm not hearing you, Brian, or seeing you. I know Zoom can be tricky for those of us who are used to Microsoft Teams.
Well, he's still muted. That's what I see.
Yeah, likewise. Okay, well, with that, I'd just like to thank you again, Dr. Hernes, for taking the time and for this really informative presentation. And to your colleagues and to the team for all of the work. So with that, we're gonna use the next few minutes just to wrap up today's seminar. And so as you can probably see already, an exit poll has popped up. So please take a few minutes to fill that out. So while you're filling that out, I will just highlight the upcoming or the next seminar. I don't know if we can go to the next slide here, but I believe the next seminar is going to be focused on collective action and co-production, especially in integrated social ecological systems. So we'll have Dr. John Takakawa from the Sassoon Resources Conservation District and colleagues who will present on their project, integrating social and ecological research to control invasive species, fostering collective action among private and public stakeholders. So we hope you can join us. We will be following up with these recordings will be posted on our website, and info or information sheets will be made available that kind of distills the takeaways from these talks. And we will do our best to follow up on the questions too that were asked that we weren't able to answer here today. So with that, unless there's anything else I'm forgetting, and others please chime in here, I suggest we wrap up. So thank you all for being here. Thank you to our presenters, and we look forward to seeing you next week at the same time. Thank you very much.
Thank you.
Thank you.